To begin this presentation, here's a brief description of the receptor. Coiled 7 times around the plasma membrane, this single polypeptide chain has a head, the end terminal on the outside of the membrane, where the large extracellular ligand binding domain is held. The tail, known as the C-terminal, is on the outside of the plasma membrane that sends a signal to the GDP binding protein receptor. Step 1. When a ligand binds to the extracellular domain of the receptor, a conformational change occurs. Epinephrine is the ligand in this process, which is mainly secreted by the medulla of the adrenal glands and functions to primarily increase cardiac output and to raise glucose levels in the blood. This signals the GDP binding protein to transfer itself next to the receptor. Structure of the G protein, or GDP. The trimeric G protein has 3 protein subunits, alpha, beta, and gamma. The alpha and gamma subunits both have covalently attached lipid tails facing the cytosolic region of the membrane that anchor it to the plasma membrane. GDP is inactively bound to the alpha subunit.
Once the receptor is activated, the trimeric G protein binds to the receptor. Then, GDP gets a signal which causes it to dissociate. GTP, abundant in the cytosol, then readily binds to the alpha subunit in place of GDP. This activates the trimeric protein to split into an alpha complex and a beta-gamma complex. Now separate, these complexes can regulate target proteins in the plasma membrane. Specifically, the alpha subunit moves to the adenylyl cyclase and activates it. Adenylyl cyclase then catalyzes the formation of CAMP. CAMP binds to the protein kinase A and activates it, allowing pKa, protein kinase A, to phosphorylate downstream factors to produce a cellular response.
The initiation of a signaling pathway results in a cellular response to changes in the external environment. This cascade occurs when pKa activates phosphorylate, and then activates glycogen, which is then converted into glucose. Ultimately, this is released into the bloodstream. The release of glucose is important, as it provides an immediate energy source for the body to aid in response to external stimuli.